So today we will talk about the link reaction. So basically in the previous video we talked about the glycolysis, and in the product of glycolysis the pyruvate was produced, if you don't remember. So watch my previous video. The pyruvic acid, two molecules of pyruvic acid were produced. So today we'll talk about the link reaction. Basically pyruvate, it in the result of the which is made in the result of glycolysis, is a polar molecule, is a polar and charged molecule, right? So it can't enter the mitochondria directly, so it cannot be entered, it cannot enter in mitochondria directly, right? So it cannot enter the mitochondria directly, so it means it has to be converted into another compound. So conversion is required, right? So what I said is basically the pyruvate is a polar molecule, so it cannot directly enter into mitochondria. So the conversion is required. We have to convert this pyruvate into something. Now what will happen, basically, like, consider that here we have pyruvate. Now what will happen, this pyruvate will be converted into acetyl aldehyde. This is converted into pyruvate is converted into acetyl aldehyde. Okay, basically this reaction is happening in the cytoplasm. This is happening, like, now the conversion is happening in the cytoplasm, right? So pyruvate is converted into acetaldehyde with the realization of one carbon dioxide, and one molecule of carbon dioxide is released. So, and the enzyme used over here is basically pyruvate. Enzyme used over here is pyruvate dehydrogenase, pyruvate decarboxylase. Why decarboxylase? Because carbon dioxide is removed. So pyruvate decarboxylase is used to convert pyruvate into acetyl aldehyde, and this reaction is now being taken place in the cytoplasm. Now what will happen? This acetyl aldehyde will be converted into acetyl. This acetyl aldehyde is converted into acetyl by the action of this converted into acetyl. This reaction is still happening in the cytoplasm. Don't look at this picture right now, right? I'll explain you. So this acetaldehyde is converted into acetyl by the action of the enzyme known as acetaldehyde, or you can say by the enzyme dehydrogenase. Dehydrogenase. And basically during this reaction, like whenever we use dehydrogenase, NAD is converted into NADH. It is converted into NADH. Right, now what is NAD and NADH? Nicotinamide dinucleotide, right? So basically pyruvate is first converted into acetaldehyde, then acetaldehyde is converted into acetyl by the action of the enzyme known as dehydrogenase, and during this reaction NAD is converted into NADH. Now this basically, and this acetyl will move to mitochondria. This acetyl will move to mitochondria. mitochondria. This acetyl will move to mitochondria, and there it will attach to coenzyme A. It will attach to coenzyme A, right? And then it will form acetyl coenzyme A, and it will form acetyl coenzyme A. So acetyl coenzyme A will be formed in the mitochondria, like in the inner membrane of the mitochondria. So basically the first two steps happens in the cytoplasm. As I said, the pyruvate is a charged molecule, so it can't directly enter, like as it's shown over here, it can't directly enter into the mitochondria. So it must be converted into acetyl in the cytoplasm. So as I said, my previous, there are two molecules of pyruvate, so two molecules of acetaldehyde and two NADH. NAD will be converted to NADH and two acetyl. So there will be two molecules of acetyl coenzyme A. So remember this thing that the acetaldehyde basically is a two carbon molecule. It is a two carbon compound. Right? Why? Because pyruvate is three carbon. One carbon is removed by the action of decarboxylase. Right? So now how many carbon left? Two carbon are left. So now this two acetyl coenzyme are formed, and now this acetyl coenzyme will go into the Krebs cycle, or you can say the TC cycle, tricarboxylic cycle, right? So here in the given diagram you can easily understand this. Don't consider this, right? Pyruvate will be converted into the acetyl coenzyme A. So now let's look at the structure of acetyl coenzyme A. So basically the structure is something like this. There is a pentose sugar. Over here we have nitrogenous base, which is adenine. This is, okay, carbon number 1, carbon number 2, carbon number 3, carbon number 4 over here, and here we have carbon number 5. So basically one phosphate is attached to carbon number 3, phosphate is attached, and one phosphate is attached to the carbon number 5. Now this phosphate is basically attached to coenzyme A and to this. Basically this is the structure. I know I haven't drawn it in the correct way, but it's something like this. I can make it in a straight manner. This on carbon number 5, a phosphate is attached, and phosphate coenzyme A is attached, and with coenzyme A, oh, there is sulfur. So basically this compound is known as, this coenzyme A is known as phosphotinic acid. Phosphotinic acid, or also known as vitamin B5, right? We have phosphopentotenic acid, which is also known as vitamin B5, and this is known as 3,5, you can say adenosine 3,5 bisphosphate. Adenosine, I can write over here, is known as adenosine 3,5 bisphosphate, right? So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section, and I'll be very happy to answer you guys, right?